Okay, so let us get started for today's session. Let me share the entire screen. Mhm. Yes. So welcome everyone to the Introduction in Developmental Biology Week 6 PMRF-NPTEL session. So as you would know, these are problem solving sessions for the NPTEL course Introduction to Developmental Biology, in which we go through the previous year's quizzes of the same course in order to solve them and get a better understanding of the concepts behind the course, as well as help us to solve different problems in the quizzes as well as the final exam for the NPTEL. I will quickly introduce myself. My name is Anuvab Chatterjee. I'm a PMRF fellow and PhD scholar at IIT Kanpur, and I will be taking this course. So how exactly do we do this course? Generally, I show you a particular quiz from the previous year's run of this course, and I ask you to try and solve the problem, and then I show you the concept behind the course. We have a discussion, and then we actually see what the correct answer is. So to start off for today's session, we are going to go right into the first question, which is choose the correct statements regarding receptor tyrosine kinase or RTK pathways. The options are: a gain of function mutation in the RAS gap may lead to cancer, a loss of function mutation in ERK will lead to excessive target gene expression, RTK signaling effects are limited to regulation of cell cycle, injecting purified GTP within the cell will cause the activation of the RTK signaling, serine is a suitable substitute for tyrosine residues within the receptor for phosphorylation, MEK is epistatic to ERK, and dimerization of receptors is required for downstream signaling. So this is a more than one correct kind of a question. So you can think and choose which of these are going to be the correct option. There are multiple here. So I will give you a moment to think about this. Okay, hopefully you have looked through it. So let us try and understand the RTK pathway first, and then we'll try and choose which of the which of the options are correct. So this is the receptor tyrosine RTK pathway. So how does it work? Screen here. So the RTK pathway is a signaling pathway. So there is obviously going to be a ligand and a receptor. So the ligand normally binds to the receptor on the cell surface like this, and this is going to cause changes in the intracellular domain of the receptor, causing multiple autophosphorylations to occur. So this receptor is a dimer, and it will start phosphorylating the serine or tyrosine residues on both arms of the ligand. These receptor intracellular domains will in turn phosphorylate and cause the signal to go downstream. So if the once the ligand binds to the receptor, the first thing that happens is that the intracellular domain gets phosphorylated, and the first protein that it in turn phosphorylates is known as the SOS or adapter protein. So this is son of 7-less in case of Drosophila, and once SOS is phosphorylated, the GNRP domain, which helps to transmit the signal further downstream, will go ahead and phosphorylate a monomeric GTP binding protein known as RAS. So normally RAS is inactive when the ligand has not bound to the RTK and is bound to a GDP molecule. The GNRP will help the GDP to be substituted to be substituted with a GTP. The RAS bound with the GTP with it is the active form of RAS. And what RAS GTP will do, it will phosphorylate the next downstream partner, which is RAF, which will then phosphorylate the next downstream partner MEC, which will finally phosphorylate ERK, which will travel to the nucleus and then phosphorylate different transcription factors which are inactive and make them into active forms, which will bind to the DNA in the nucleus and cause transcription, and essentially the genes will be transcribed which are activated by the pathway. So here it is a straightforward form written as words. So ligand binds to the RTK, RTK phosphorylates the GNRP, GNRP the RAS, then the RAF, then the MEC, the ERK, the transcription factor, followed by transcription. So this is essentially a signaling cascade in which the signal is amplified intracellularly, because there is always going to be more the RAS is going to phosphorylate a lot more RAF, the RAF will phosphorylate a lot more MEC, MEC will phosphorylate a lot more ERK. So essentially the signal is amplified because of the abundance of the molecules downstream, and finally we have transcription. And there is a regulation step involved in this as well. One of the intracellular domains activates the entire pathway, whereas the other domain actually phosphorylates and activates the gap proteins. What the gaps do is that they go ahead and dephosphorylate the GTP bound to RAS. Once this RAS GTP is dephosphorylated, it goes back to its inactive RAS GDP form. So essentially you have a regulation here. So more gap, less activation. So you essentially control the entire pathway so it does not run free. So once you activate it, it does not cause all the genes to be transcribed all the time. The gap will cause a regulation to occur. And there are different functions that the RTK pathway does. In C. elegans, we have looked at it. It helps in vulva development. We have been looking at it in the past few weeks. In Drosophila, it helps in eye development, and in humans, it causes cell proliferation. So this is essentially the pathway. And if we look at the different options, the first is a gain of function mutation in RAS gap may lead to cancer. So what is the RAS gap? This is this protein. So more and more, say if there is a gain of function mutation of gap, that means that without even the ligand binding, gap will always be active. And if gap is always active, RAS cannot stay in the RAS GTP form. It will always be in its inactive form, so it actually down regulates the entire signaling. So gain of function mutation in RAS gap may lead to cancer. That is obviously not correct, because cancer would need activation of the entire pathway, and because it causes self proliferation, more cell proliferation, so it will lead to cancer. But with the gain of function in RAS gap, it will actually down regulate and cause less cell proliferation, or actually completely stop the pathway, and thus this option will not be correct. A loss of function mutation in ERK will lead to excessive target gene expression. So if you look at it again, ERK is the final downstream target that goes into the nucleus and activates transcription. So a loss of function of ERK will essentially remove this. So even if the RTK pathway is active with the ligand binding to the receptor, there will be no downstream transcription, and so no ERK being there will cause no transcription. And the option says will lead to excessive target gene expression, which is obviously not true, so this is also not a correct option. RTK signaling effects are limited to regulation of cell cycle. That is not true, because we have been looking at it that it helps a lot in vulval development via the anchor cell communicating to the P6P and other equivalent cells in the primordia of the vulva, and it does many other things like eye development and stuff. So not true. Injecting purified GTP within the cell will cause activation of RTK signaling. So GTP binding to RAS actually activates the pathway, but just having more GTP won't help, because there needs to be a partner GnRP, which is only activated via the ligand binding to the receptor, which helps the substitution of the GTP to the GTP. So even if you put in more GTP, it will not activate the pathway without the ligand binding. Serine is a suitable substitute for tyrosine residues within the receptor for phosphorylation. This is indeed true. So these are known as serine. So these tyrosine path, the amino acid tyrosine are the ones that are being phosphorylated, but serine can be substituted in their place to actually cause the phosphorylation instead, or be phosphorylated in turn, because they have the same OH group at their ends, if you remember your amino acid biology. So they can actually do this. Then MEC is epistatic to ERK. So where is MEC? MEC is upstream to ERK. MEC is upstream to ERK, and if we remember that in case of sequentially activating enzymatic pathways, whichever gene is upstream is called the epistatic gene, because without MEC there is not going to be any activation of ERK. So in the basic epistatic pathway, if you remember, so this we have dealt with a bit earlier, but just to refresh you, so if you do not know what order a particular enzymatic pathway works in, so if MEC is actually upstream to ERK, or ERK comes first and then MEC comes, you make mutants of both. So you knock out MEC, you knock out ERK, and then you also do something known as a double mutant. You knock out both MEC and ERK. Whichever single mutant has the same phenotype as the double mutant is known as the epistatic gene and is placed upstream. So just for example, if you knock out both MEC and ERK, you actually stop the entire pathway at phosphorylated RAF. And again, if you just knock out MEC, you again stop the entire pathway as phosphorylated RAF. So if your readout is phosphorylated RAF, then both the single MEC mutant and the double mutant have the same readout, phosphorylated RAF, and no downstream gene transcription. So double mutant has the same phenotype as the single mutant. So MEC is epistatic to ERK and is actually upstream. This is how essentially most of the genetic pathways, the enzymatic pathways were figured out, and we got to know that what comes first. So MEC is epistatic to ERK. Just looking at the pathway, whichever comes first is actually epistatic. That is what we call it. So MEC is epistatic to ERK. So this option is also true. And finally, dimerization of receptors is required for downstream signaling. So once the ligand bind, it leads to receptor dimerization, which then in turn leads to autophosphorylation. So dimerization is important, and that only happens when the ligand binds. So dimerization of receptors is required for downstream signaling. So all these options are the correct ones. So any questions, then you can always put it into the NPTEL forum or the YouTube chat, and then we can have a further discussion in case this is not clear. Okay. Going forward, we are talking about different pathway known as the JAK-STAT pathway. So in this pathway, phosphorylated STAT binds and activates transcription factors responsible for gene expression. Activated JAK receptors activate STAT by phosphorylating tyrosine residues. Dimerized STAT enters the nucleus and regulates gene expression. Loss of function mutations in FGFR3 or FGF receptor 3 result in failure to activate STAT1, signaling in thanatophoric dysplasia, or a gain of function mutation in the kinase domain of JAK receptors will have the same phenotype as the loss of function mutation in the DNA binding domain of STAT. Again, this is a more than one option correct type of a question. So, you have to think about it and choose more than one option which you think is correct. I will give you a moment to think about it. Okay, let us look at the JAK-STAT pathway. This pathway is a variation on the RTK receptor pathway but works very similarly and helps in differentiation of blood cells, growth of limbs, activation of casein during milk production. So how does it work? The starting is almost the same. The ligand binds and causes dimerization of receptors, which causes transphosphorylation and activation of a downstream target known as JAK or Janus kinase. Janus kinase gets phosphorylated, as we saw GNRP getting phosphorylated in the last one. But in this case, what JAK does, it causes phosphorylation of a downstream partner course known as STAT. Once STAT is phosphorylated, it does something interesting. Two phosphorylated STAT molecules come together and dimerize like this, and the dimerized STAT can enter into the nucleus directly and act as transcription factors. So they do not they do not activate a transcription factor downstream, but cause direct phosphorylation and cause direct transcription downstream. So this is pretty small pathway compared to everything else. So ligand binds to receptors, which causes phosphorylation of JAK, phosphorylation of STAT. STAT dimerizes, goes into the nucleus and activates transcription. Pretty straightforward. And there is this pretty important disorder that occurs in case there is a problem with the JAK-STAT pathway. So the JAK-STAT pathway is involved in chondrocyte proliferation. So what is chondrocyte proliferation? So when our long bones form, so the limb bones and ribs and everything else forms, they actually first are like a ball of cells which are called chondrocytes. These chondrocytes are present as a ball of cells towards the where the limb is going to form, and they proliferate a lot and essentially make a framework of the entire limb, and slowly they change into bone. So that is how the limb forms. There is a chondrocyte population which proliferates, lays down the framework of the entire limb, and slowly these chondrocytes change to bone cells, and you have the entire bone forming. But in case and the JAK-STAT pathway actually helps in chondrocyte proliferation. So it causes the limbs to actually grow to their full length. But in case of a particular mutation in one of these JAK-STAT pathway, in which the receptor is the FGFR3 receptor, there is a mutation which causes a gain of function mutation. So what does it do? There is a mutation in the extracellular domain which causes this entire pathway to be always active, even though there is no uh, yeah even though there is no ligand binding, there will always be a gain of function here. So the pathway is always active even if there is no ligand binding. So what this causes is that it always causes STAT to get phosphorylated. So essentially there is no need for a ligand to bind here, because the receptor is always active. It always causes the STAT to dimerize and go into the nucleus and start transcription, and this causes premature suppression of chondrocyte proliferation. So the framework of the chondrocytes that actually will make the bones the correct length does not occur. Cartilage stops growing, and a smaller cartilage framework converts into bone. So here you have shortened limbs, shortened ribs. Essentially all the bones, they are shorter than they should be, and this is known as thanatophoric dysplasia, in which you have narrow chest, extremely short limbs, and this is fatal. So what happens? This JAK-STAT pathway via the FGFR3 receptor helps in cartilage, so it stops cartilage proliferation at one point, but due to the mutation it is always active and it stops cartilage proliferation at a very early stage. The entire cartilage primordia doesn't grow to the correct length to which the bones should form, and they form shorter bones, shorter ribs, and this causes a lot of problem. Essentially causes the ribs to be really small, and like the entire rib cage to be too small for the lungs to actually breathe, and thus this entire thing is fatal, which is thanatophoric dysplasia. So let us look at all the options now. The first option is phosphorylated STAT binds and activates transcription factors responsible for gene expression, but we have just seen that phosphorylated STAT is in itself a transcription factor. It does not activate anything, goes to the nucleus and does its job by itself. So this option is not true. Activated JAK receptors activate STAT by phosphorylating tyrosine residues. This is true. STAT is phosphorylated at the tyrosine residues. Dimerized STAT enters the nucleus and regulates gene expression. True again. Loss of function of FGFR3 results in failure to activate STAT1 signaling in thanatophoric dysplasia, which is not true. We just saw that it is a gain of function mutation which always activates STAT1, which causes premature suppression of chondrocyte proliferation, which causes thanatophoric dysplasia. So this option is not correct. And a gain of function mutation in the kinase domain of JAK receptors will have the same phenotype as a loss of function mutation in the DNA binding domain of STAT. So look at it carefully. Gain of function in the kinase domain of JAK is equivalent, according to this option, to a loss of function mutation in the DNA binding domain of STAT. So let us look at the pathway again. So a gain of function mutation in JAK, what it will do is that even with no ligand binding to the receptor, it will be always phosphorylated or act as if it is phosphorylated, and then so essentially the entire pathway is always active, because JAK, if it is phosphorylated, it will cause STAT dimerization and activation of downstream genes. So gain of function mutation of JAK equivalent to activation of the pathway, but loss of function of STAT5 DNA binding domain, what will that do? So in case of a transcription factor, there are two important domains that you should always know about. The first one is known as a DNA binding domain, which helps the transcription factor to recognize where it should bind and go and bind to the DNA. And there is a trans-activating domain, which is another different domain which actually helps it to cause the gene transcription to occur by mostly activating the RNA polymerase II and actually pushing it along and causing it to polymerize the mRNA. So two important domains in a transcription factor. First, the DNA binding domain identifies and binds to the DNA. Trans-activating domain, which actually causes the transcription to occur. So dimerized STAT5 is a transcription factor, and a loss of function in the DNA binding domain of STAT5 will cause it not to bind to the DNA at all, and thus even though there is full-on activation upstream, because STAT5 cannot bind to the DNA, it will not activate transcription. So a loss of function of DNA binding domain of STAT5 will cause no gene transcription, and hence the entire pathway will be shut down. So STAT5 loss of function of the DNA binding domain will actually cause the entire pathway to not work. So going to the option, it says that the gain of function mutation in the kinase domain of JAK receptor will have the same phenotype as a loss of function mutation in the DNA binding domain of STAT, which is false, as we just discussed. Gain of function mutation in JAK will cause activation of the pathway, and loss of function mutation in the DNA binding domain of STAT will cause a inactivation, deactivation of the pathway. So this option is also not correct. So these two are the correct options. Okay. That is all well. We move forward to the next question. Which of the following is true regarding the Hedgehog signaling pathway? The options are: deficiency in palmate synthesis can interfere with Hedgehog signaling. The protein dispatched is required for secreting membrane-enclosed Hedgehog. Hedgehog is secreted as lipoprotein assemblies with the help of heparin sulfate proteoglycans. Cholesterol is required for receptor binding of Hedgehog, and loss of function mutation in patched will have the same effect as the gain of function mutation in smoothed. So again, I will give you a minute to figure out what you think is the correct option. Okay, so let us look now at the Hedgehog signaling pathway. So Hedgehog signaling pathway is one of the other important toolkit signaling pathways that help in development throughout, uh, and these are, you will always see, these are usual suspects in different parts of the developing embryo, which helps in the essentially morphogenesis. So we are talking about the Hedgehog signaling pathway. So how does Hedgehog ligand is a bit different and interesting compared to the other ligands, which are mostly proteins, with like hydrophilic proteins rather. So how does the Hedgehog ligand look like? So Hedgehog ligand actually forms as a precursor protein first. So it's a signal sequence, a signaling domain, and an auto-processing domain in the entire precursor protein. What happens is that the auto-processing domain can do something known as a self-cleavage. It actually just cleaves itself off. So it actual ligand is very small. Further, sonic hedgehog has two interesting post-translational modification. First being a cholesterol modification, which is added to the C-terminal end, and a palmitate addition or palmitylation at the N terminal. So essentially you add two huge lipid moieties to the protein, causing it to be very hydrophobic in nature, because lipids do not like water, and interestingly the entire extracellular space is full of water. So hedgehog needs to be very well uh, handle, because otherwise the hedgehog will not be able to at all travel the extracellular space, because everything is hydrophilic and it is highly hydrophobic. The first thing that occurs is that hedgehog, this active hedgehog which has the palmitate and the cholesterol added, is transported out of the cell using the transmembrane protein dispatched. So essentially it dispatches hedgehog, and that it does by attaching it to different lipid moieties. So essentially it forms different, so it can throw hedgehog out as a monomer, but this monomer quickly settles down close to the cell because it cannot travel outside in the hydrophilic environment. Actual signaling, or rather longer range signaling, occurs by these forms of hedgehog, in which the hydrophobic parts of the entire protein is somehow hidden from the hydrophilic extracellular environment. This can either be as a hedgehog lipoprotein, in which, as you can see, all the hydrophobic regions are inside, hedgehog multimer, in which again it is towards the inside, so that the hydrophobic region is protected from the extracellular hydrophilic environment, or as a nodal vesicular parcel, essentially helping, like it takes a part of the membrane with it, which covers it so that it is again protected from the extracellular hydrophilic environment. There is another way this occurs, in which it attaches to a carrier protein known as COOB2, which actually holds on to the hydrophobic part of the hedgehog ligand, and it holding on to the hedgehog ligand, the hydrophobic part of the hedgehog ligand actually hides that part. So that Scoob hedgehog combination can actually do longer range signalings because now it can travel in the hydrophilic environment. So essentially, hedgehog, for it to be active signaling, it needs to have a palmitate and a cholesterol added, and extracellular signaling occurs when the dispatched protein, which is a cell surface molecule, helps it get out of the cell in one of these protected forms so that it can go and do its signaling part. And this is all about secretion, what happens when it actually reaches its target. So hedgehog binds to a particular receptor known as the patched protein. Let us first look at how it occurs, like behaves when hedgehog signaling is inactive. So when there is no hedgehog around, the patched actually goes ahead and inhibits another protein, which is a surface protein known as smoothant. So no hedgehog, patched clamps down on smoothant and causes it not to do its function. When this entire complex is inactive, we have a particular transcription factor called CI, which stays in the cytoplasm and is cleaved by protein kinase A. Once it is cleaved, part of the CI complex goes into CI protein goes into the nucleus and acts as a repressor. So no hedgehog, CI gets cleaved, goes to the nucleus, acts as a repressor. But the moment hedgehog is there, smoothant, the patch no longer clamps down on smoothant and causes it to be inactive. And once smoothant is active, it inhibits both pKa and slim, which together cleave CI. If both of them are now inactivated because of smoothant, CI, the whole protein can now go into the nucleus and cause transcriptional activation. So essentially, no hedgehog, CI gets cleaved, repression. Hedgehog, CI does not get cleaved, entire thing enters the nucleus, activation of genes. This happens really interestingly in so this entire thing occurs was figured out in Drosophila, but in higher eukaryotes you actually see the primary cilia doing a lot of things. So smoothant, instead of being associated with patched, is essentially translocates to this organelle called the primary cilia, which is a signaling organelle present on cells, and it is present in the cell surface. Smoothant enters this complex when it is not deactivated by patched, and then essentially the same cascade occurs, but the protein names are different, which are the eukaryotic, higher eukaryotic versions. So how does the hedgehog pathway work again? Hedgehog actually binds and inactivates patched from inactivating smoothant. So a double inhibition kind of a situation. When smoothant gets freed from patches, the patched proteins influence, it goes and makes sure CI is not cleaved, and CI goes and does transcription. So let us look at it. What the options are. Which of the following are true regarding hedgehog signaling? First, deficiency in palmate synthesis can interfere with hedgehog signaling, which is true. As we see, palmate and cholesterol both being added to sonic hedgehog actually makes it a signaling ligand. Without palmate addition, there will be no signaling, even though hedgehog is secreted and everything, and thus deficiency in palmate synthesis can cause problems with it. Palmate addition also causes helps in secretion of hedgehog, so there is that as well. So this option is true. The protein dispatched is required for secreting membranous enclosed hedgehog. True. Hedgehog is secreted as lipoprotein assemblies with the help of heparin sulfate proteoglycans. This is also true. As we saw, there are different lipoprotein assemblies in which hedgehog can be secreted as, and they associate with heparin sulfate proteoglycans because of that. In order to do that, uh, cholesterol is required for receptor binding of Hedgehog, which is also true, and loss of function mutation in patched will have the same effect as a gain of function mutation in smoothened. So going back here, patched holds on to smoothened and causes the entire pathway to be inactive. Loss of function of patched will cause smoothened to go haywire and do whatever it wants, which is activation of the entire pathway. So that is true. And similarly, a gain of function of smoothened, which means that even though there is no Hedgehog binding to patched, smoothened will still activate the entire pathway, which will have the same effect of activating the entire pathway. So loss of function of patched and gain of function of smoothened will have the same overall phenotype. So loss of function mutation in patched will have the same effect as gain of function mutation in smoothened. So all the options were true. Really easy. Going forward, here is the next question. Two cell lines were cultured in the presence of ligand specific to a given signaling pathway. Based on the following graph, identify the correct statements relevant to signaling required for a TCF mediated transcription of gene X. So this is the graph. We see that in the control, with the ligand being added to the entire substrate or the culture media, we have that the control cell line just shows more and more activation of gene X, whereas the test never shows any activation. So what might be the reason? That is the question we are being asked. The options are: test cell line may have a gain of function mutation in protein kinase A. Test cell line may have a loss of function mutation in smoothened. Test cell line may have a loss of function mutation in disheveled. Test cell line may have an immature stop codon in axin gene, and test cell line may have a gain of function mutation in GSK3. So I will give you a minute to figure this out. Okay, so let us look at it. So here we have been specified that the gene X is activated by a TCF mediated transcription. So let us look at what actually is TCF mediated transcription and which pathway is it part of. It is part of the Wnt signaling transduction pathway. Specifically, the canonical Wnt signaling, which was the first discovered Wnt signaling, but Wnt signaling can act in three different pathways, and we'll look at all of them. So how does Wnt signaling occur? So first I will discuss if there is no Wnt, what happens. If there is no Wnt binding to its receptor frizzled, then frizzled will go ahead and catch hold of a cell surface associated protein known as disheveled. So once disheveled is held onto by the frizzled protein, it does not do its normal function. So its normal function is inactivating something known as the degradation complex. The degradation complex is a huge complex made out of multiple protein, which include GSK3, APC, axin. So these guys are essentially people who will go ahead and degrade a specific protein, which is beta catenin. So beta catenin is always degraded in the cytoplasm by this degradation complex. But once Wnt binds to frizzled, it actually causes the disheveled to be free. Disheveled goes ahead and inactivates or suppresses the degradation complex, causing beta catenin to actually be released. Released beta catenin enters the nucleus, where it binds to left TCF transcription factors, and both of these go ahead and sit on the DNA and cause gene transcription. So Wnt binding to frizzled causes suppression of disheveled to be lifted. Disheveled in turn inhibits the degradation complex. Beta catenin is no longer degraded. Intact beta catenin enters the nucleus, binds to left TCF, goes to the DNA, binds to it, causes gene transcription. This is how the TCF mediated pathway occurs. So this is the Wnt beta catenin canonical pathway. But since we are here, we will also discuss the other ones. But this is essentially what the question is talking about: the Wnt beta catenin canonical pathway. The other ways Wnt signaling occurs is where there is no beta catenin involved but other downstream partners. So when Wnt binds to frizzled and there is a co-receptor known as STBM, it again causes disheveled to be activated. But in this case, when disheveled is activated by both the action of frizzled and STBM, it associates in a big complex which activates the monomeric GTPase rho A, which activates ROCK, which causes cytoskeletal changes, even though there are no gene transcription effects of this arm of the Wnt pathway. The other way disheveled can act is it interacts with RAC, activates it, which in turn activates either JNK, causing gene transcription, or directly again cytoskeleton changes. So this Wnt non-canonical pathway helps mostly in cytoskeletal rearrangements in the cell. And finally, the third way Wnt can interact with frizzled is that it activates via other G proteins the phospholipase C, which causes finally via cascade release of calcium. This release of calcium activates calcineurin and NF-AT, another transcription factor which goes into the nucleus and causes transcription. So these are the three main ways in which Wnt signaling works. But for this question, we are mostly interested in the canonical Wnt beta catenin signaling, in which beta catenin is the transcription factor which goes and binds to left TCF and causes transcription, and it is degraded with the degradation complex when there is no Wnt associated with frizzled. So what do we know? We know that TCF mediated gene transcription of gene X is inhibited in the test cell line. So we are looking at anything that will cause a loss of function of the entire pathway or inhibition of the pathway. So test cell line may have gain of function mutation in protein kinase A. Protein kinase A is not involved in the TCF mediated transcription of gene X, so we don't care about this option. This was in the smoothen pathway, if you remember, or the sonic hedgehog pathway. Test cell line may have a loss of function in mutation in smoothened. Again, hedgehog pathway, we don't care about this one because we are looking in TCF mediated transcription, so Wnt signaling pathway partners are what we care about. Test cell line may have a loss of function mutation in disheveled. So if there is a loss of function mutation in disheveled, the degradation complex is always active, irrelevant if the ligand and the receptor bind to each other, and thus degradation complex will always degrade the beta catenin, and so we have no activation of the pathway, which actually works for us because this is what we are looking for, and so this is the correct option. Test cell line may have an immature stop codon in axin gene, essentially causing a loss of function of axin because there is an immature stop codon in the axin, and immature stop codon in axin will cause the entire degradation complex to not be active. If there is inactivation of the degradation complex, so irrelevant if Wnt binds to frizzled, beta catenin will always enter into the nucleus and cause activation of gene transcription. So this option says that will entail that the entire gene X is always transcribed, so activation of the pathway, but this is not what we see, so this is not also a correct option. And finally, test cell line may have a gain of function mutation in GSK3, which means even though Wnt and Frizzled are bound to each other, GSK3 will always go around and cause degradation of beta catenin, so loss of the entire pathway, which is what we see here. So the last option is also correct. Okay, so these were all the correct options, and we move on to the next question. We'll deal with a different signaling pathway now, which is the TGF-beta signaling pathway. Which of the following statements regarding TGF-beta signaling pathway are correct? The options are: ligand binds to the receptor dimer to activate downstream signaling. Type 1 receptor phosphorylation occurs on serine threonine residues. SMAD4 acts as a co-SMAD that binds to all receptor activated SMADs. SMAD1 and 5 are phosphorylated by type 1 TGF-beta receptor, and SMAD2 and 3 are phosphorylated by type 1 TGF-beta receptor. So again, more than one option correct type of a question. So you have to go ahead and choose what you think is the correct option. Okay, so let us look at the TGF-beta pathway. So TGF-beta is part of a huge superfamily of ligands, and these are like a lot of ligands are present which activate similar pathways. So we are going to deal with the major two, which is the TGF-beta and activin, or the BMP ligand pathway. So how does it occur? So TGF-beta pathway receptors are serine threonine kinase domain receptors, and there are two: receptor type 2 and receptor type 1. But how does it work? When TGF-beta like ligands are around, they bind to receptor type 2, which dimerizes, and essentially so two receptor type 2s come together, and they in turn and this binding to two receptor type 2 uh, receptors with the ligand causes it to go ahead and find a receptor type 1, and which also dimerizes with the same molecule. So now there are two receptor type 2s and two receptor type 1s, so we have a huge complex which is the tetrameric receptor complex. So two receptor type 2s, two receptor type 1s, which only tetramerize in the uh, presence of a ligand binding together. So this is what happens. So now once the ligand binds to this huge tetrameric complex, the receptor type 2 does something known as transphosphorylation. So it goes ahead and phosphorylates the receptor type 1s in the GS box. This essentially activates the receptor, which causes downstream signaling. And how it occurs depends on what kind of pathway we are talking about. In case of an activin and a TGF-beta ligand, this transphosphorylation causes phosphorylation of a downstream partner, which is SMAD2 or SMAD3. So you have downstream activation of SMAD2 and 3, and SMAD2 and 3 bind to SMAD4, which is known as the co-SMAD, and together SMAD2, 3 and 4 go into the nucleus and cause gene transcription. But there is the other family in the TGF-beta pathways, which are known as the BMP ligand family, which is also huge and does a lot of different things. But the activation of the receptor complex is the same: tetramerization of the receptor which causes transphosphorylation and activation of receptor 1, and this for transphosphorylation of receptor 1 causes phosphorylation of downstream partners SMAD1, 5 or 9, and these bind to again the co-SMAD SMAD4, goes into the nucleus, causes gene transcription. So essentially the pathway is pretty simple. Ligand binds, it causes transphosphorylation and activation of the receptor. Receptor then phosphorylates either SMAD2/3 or SMAD1/5 via the receptor 1 type 1. So this type 1 is the one that phosphorylates the downstream targets, whereas type 2 transphosphorylates the type 1. And SMAD2/3 or SMAD1/5, once phosphorylated, binds to SMAD4, goes into the nucleus, transcription. And SMAD4 essentially bound to SMAD159 or SMAD2/3 act as a transcription factor and can directly initiate transcription. They do not pass on the signal to a downstream transcription factor. So that was a lot, but yes, so that is how the pathway works, and let us look at it. What the options are: ligand binds to receptor dimer to activate downstream signaling. That is false, it is a tetramer. Type 1 receptor phosphorylation occurs on serine threonine residues. That is true. TGF-beta receptors are serine threonine kinases, so essentially they are phosphorylated and phosphorylate. They are phosphorylated on the serine threonine amino acids. SMAD4 acts as co-SMAD and binds all receptor activated SMADs. That is true. SMAD4 is the downstream target for all SMADs, and all activated SMADs bind to SMAD4 to go into the nucleus. SMAD1/5 are phosphorylated by the type 1 TGF-beta receptors. That, as I said, is true. The type 1 receptor are the one that actually phosphorylate the downstream partners, not the type 2. So this is true, and SMAD2/3 are phosphorylated by the type 1 TGF-beta receptor, which is also true. So essentially all the SMADs other than SMAD4 are phosphorylated by the type 1 TGF-beta receptor. Okay, then go on to the next one, and we go on to a different pathway, which is the apoptosis pathway. The question is, choose the correct statements during apoptosis. The options are: overexpression of the protein BAX may prevent cell death. If APAF1 has a nonsense mutation in one allele while the other allele is wild type, it will function normally. A gain of function mutation in BCL2 may prevent cell death. Functional caspases are developmentally more important in mammals compared to C. elegans. In C. elegans, Eagle1 overexpression may promote apoptosis. So choose whichever you think is the correct option. Again, this is more than one correct type of a question. Okay, so let us get back to the question. Again, the question was, choose correct statements regarding apoptosis. Overexpression of BAX may prevent cell death. If APAF1 has a nonsense mutation in one allele while the other allele is wild type, it will function normally. A gain of function mutation in BCL2 may prevent cell death. Functional caspases are developmentally more important in mammals compared to C. elegans. In C. elegans, Eagle1 overexpression may promote apoptosis. So what is apoptosis? Apoptosis is programmed cell death, as we all know. So This was first discovered in C. elegans development, in which they figured out that some cells developmentally are always destined to die, and they are programmed to die, and that is why it is known as programmed cell death. So how does the programmed cell death works? It starts at in case of C. elegans, starts all the way over at the mitochondria. In the mitochondria, there are on the surface of the mitochondria there is a complex which is composed of SED9 and SED4. When apoptosis has to occur, the cell expresses a lot of Eagle-1 protein which displaces SED9 with Eagle-1. So the SED4 goes ahead and forms a complex by itself because there is a lot of SED4 non-associated SED4 around. SED4 then binds to SED3, and both of these together go ahead and act. Once this complex is assembled, this causes the SED3 to be active, and this SED3 protease is known as the executioner protein, which goes ahead and chops everything up in the cell and causes apoptosis. Here we have an example in which C. elegans, this is the C. elegans gonads, and here we have particular cells which are button shaped. These are undergoing apoptosis. So in C. elegans it is pretty straightforward. Eagle-1 inhibits SED9. SED9's normal function is to catch hold of SED4 and inhibit SED4, but with Eagle-1, a lot of Eagle-1, SED9 is cannot do its normal function. SED4 is free. SED4 activates the executioner SED3. SED3 causes apoptosis to occur. In case of mammals and especially mammalian neurons, uh, the in case of SED9, the SED9 and SED4 being partners, BCL2 and APAF1 are the partner molecules. And again, the molecules which cause apoptosis to occur are known as BIC and BAX. BIC and BAX being in the cytoplasm go ahead and inhibit BCL2. BCL2 being inhibited releases APAF1. APAF1 activates caspase-9, which is equivalent to SED3. Caspase-9 actually activates a second caspase, caspase-3, which is the executioner protease, which chops everything up, and you have apoptosis. Okay, and in case of worms, C. elegans, they figured out like apoptosis is important, and there are like 100 plus cells which apoptosize for normal development of the C. elegans worm. But even if apoptosis was inactivated, so you say inactivate the executioner caspase SED3 or protease SED3, the cell is, the worm is completely viable and does its normal function, develops normally, goes about its job. There is no as such problem with the worm. But in case of mammals, this is a huge thing, that if apoptosis is inactivated by loss of function of caspase 3, say, then or caspase 9 in this example, so this is a normal wild type embryo, you see it is normally developed, but in case of caspase 9 knockout, so there is no apoptosis occurring, you see there are a lot of problems. There are multiple neurons in the brain which are programmed to die, and with them not dying off, the brain is malformed. The fingers, they have the webbing in them, so the fingers are not disjointed at separate fingers but are together because the intra-digital mesenchyme has not apoptosized. So essentially this entire mutant is not viable. And a cross section of the brain also shows that in case of wild type, you have these vacant spaces which are called ventricles, and you have normal formation of the brain, but in case of the knockout, there is overgrowth of neurons everywhere and the ventricles are not even visible, so not viable. So this is apoptosis, and in normal development you absolutely require apoptosis to work well for viable normal embryos. And now let us look at it. The first option, so let us look at the question now. The first option is overexpression of BAX may prevent cell death. So as we saw, BAX, actually more BAX causes inactivation of BCL2 and promotes apoptosis. So the first option is not correct. If APAF1 has a nonsense mutation in one allele while the other allele is wild type, it will function normally. Mm, this is just not true. There is not enough information. This is also not correct. Gain of function mutation in BCL2 may prevent cell death, which is correct. BCL2 is anti-apoptotic, as we saw. It holds on to APAF1 and prevents the apoptosis to go forward. So this is the correct option. Functional caspases are developmentally more important in mammals compared to C. elegans. As we saw, C. elegans doesn't care if apoptosis does not occur. It can go around its way very well, but in case of mammals, it is especially important because the embryo will just be not viable. And in C. elegans, Eagle1 overexpression may promote apoptosis. So we know in C. elegans, if there is a lot of Eagle1, it will displace SED4 from SED9 and cause apoptosis. So Eagle1 is pro-apoptotic, and overexpression will indeed promote apoptosis. So this is these are the correct options. Moving forward, identify the correct signaling interactions in this image. So this is us going all the way back to C. elegans vulva development. So these are the four options that we have shown. That is this how signaling works, and you have to choose which ones are correct. In A, we see anchor cells sending out some signals to all the cells, but majorly to the P5, P6, P7P cells. In B, it sends out signals only to P5, P6 and P7P, but most of the signal is to P6P. In C, P6P is releasing ligands, Lin12, and P5P is accepting the ligands. And in P6P, it is essentially uh, not releasing ligands, but this is juxtacrine signaling, in which P6P has surface associated signaling molecules which bind to P5P, the receptors on P5P. So a lot of options, you have to choose which one is correct. This is only one option correct kind of a question. So go ahead and choose what you think is the correct option. Okay, so let us look at it. So going all the way back to C. elegans vulva development, let us just have a quick refresher. So in C. elegans vulva development, there are all of these cells which are known as which are the P6P, P7P, P8P, P3P, all the 3, 4, 5, 6, 7, 8 cells here, and these cells will either have a primary, secondary, or tertiary fate and produce some structures which only that kind of a cell can produce. Normally, there is a cell which is present on the gonad known as the anchor cell, which sends a RTK pathway kind of a signal to the cells in the middle, mainly the P5, P6, and P7P. But since the P6P is in the middle and closest, it receives the most of the signal, and has a primary fate and causes the vulva primary for structures of the vulva to form. And now P6P, since again it has the most of the signal from anchor cell, does something known as juxtacrine signaling, especially the notch delta pathway, where it signals P5, P and P7P to not become a primary but a secondary cell, and have a secondary cell fate and cause form other structures in the vulva. So essentially P5P, P6P, all of these cells could have actually formed any of the fates, primary, secondary or tertiary. But the signal from the anchor cell causes only the cells which are closest, P5, P6 and P7P, to have either primary or secondary fate, and the cell which receives the most of the signal actually asks the other ones via juxtacrine notch delta signaling to only have the secondary fate. So this is what we see. P6P always forms the primary structure, the central part of the vulva. P5P and P7P form the lateral parts of the vulva, and all the others, P3P, P4P and P8P, do not receive any signal from the anchor cell and form tertiary structures, which are essentially the hypodermis surrounding the vulva. So there is both RTK pathway and notch delta juxtacrine pathway going on here, and this gives us a good moment to actually discuss what is notch delta and juxtacrine signaling. So notch delta signaling is a juxtacrine signaling. So juxtacrine signals occurs in cells which are in close contact to each other, where the receptors and the ligands both are present on the cell surface. So no secretion of ligands, no short range or long range signaling, but just cells attached to each other. They signal via essentially a handshake between the receptor and the ligand, which are bound to both the cell membranes of the two cells. So here the ligand is essentially known as delta, and it binds to receptor notch, which is present on the other cell. Once this binds, there is a protease which becomes active and cleaves off the intracellular part of notch. An intracellular part of notch travels all the way to the nucleus, binds to other protein and causes gene transcription. This is essentially how notch delta signaling works. Delta binds to notch, part of it gets cleaved off, goes into the nucleus, transcription. As easy as that. But notch delta signaling is actually very important in particular contexts, especially as we saw in case of our vulva development. So it actually between equivalent cells which can form similar structure, as we saw in the case of vulva development, everyone could have been primary, especially the cells in the middle, the P5P, P6P, P7P, all receive the RTK signaling. But notch delta signals from all the cells actually force some of them to form something specific. So we are discussing this in case of two other cells, which are the anchor cell and the ventral uterine precursor cells. Originally these cells are Z1PPP and Z4AAA, which when they have not matured, both of which can either form the anchor cell or the ventral uterine precursor cell. So it is by random chance that one forms one or the other forms the other one. But what decides that, even though it is random chance? It is notch delta signaling. So originally both the cells produce equal number of delta ligand, which is this bulb thingies, and the receptor, which are these hollow U's which describe the receptor. So normally these are almost, because the cells are equivalent, they produce the same amount which interact with each other. But by random chance, one of them will start expressing a bit more ligand and the other one a bit more receptor. Just by random chance, like it might happen that one of them start expressing more ligand than the receptor, and once that happens, it will cause a feedback effect. The cell which produces more and more of the ligand will ask the other cell to produce more and more of the receptor, and vice versa, till one cell is only expressing the ligand and the other cell is only expressing the receptor. And this will cause the cells to actually change their fate and decide to be one type of cell or the other. So in our current conversation, between the Z1 PPP and Z4 AAA cells, whichever produces ligands more and whichever produces only receptors, those will form the anchor cell and the ventral uterine precursor cell respectively. Similar thing occurs in mammalian neural development when there are a lot of equivalent cells where all of the cells will do the same thing, produce same amount of receptors and same amount of ligands, but by random chance one of the cells will be producing less ligands and more receptors, and via this same specific feedback loop they will be asked to produce more and more of the ligands and not receptors, and finally they will only produce ligands and no receptors, and that will cause them to convert from a neural fate to a glial fate. They will now form a glial cell instead of a neural cell, and but essentially the cells could have formed either neurons or glia, but via the notch delta signaling and essentially random chance they are specified to form one or the other. Okay, so this is what it is. So model for generation of two cell types by notch signaling. Initial difference between equivalent cells are created by random chance. This initial difference is reinforced by feedback and causes them to be determined to form a particular cell. Okay, now looking at the options, we have seen entire vulva development. The first thing shows that the anchor cell is sending out signals to every one of the cells. That is not true. It only sends out signals to the three in the middle. So B is the correct option. P5, P6, P7, P only receive signal. C is P6P secreting ligands, which P5P accepts. That is not true. It is notch delta signaling or juxtacrine signaling. So they essentially they produce receptors and ligands on the surface and do a kind of a handshake like we see in D. So the correct option is B and D. Okay. Moving on. Identify the correct statements concerning the role of extracellular matrix in development. The options are: a block in carbohydrate synthesis may adversely affect morphogenesis. A defect in fibronectin synthesis may adversely affect gametogenesis. Tissue scaffold treated with calcium chelators will prevent cell adhesion. Integrins are ECM components required for relaying signals to the cytoskeleton components. Cancer metastasis requires expression of ECM degrading enzymes. Okay, so let us look at it. So extracellular matrix. So cells in our body do not actually just they are just it is not that they are just cells in our body. The cells actually stand on top of a matrix that they secrete, which is like a mat or essentially a floor on which the cells sit. And this so this is electron microscopy image of this thing. So here on top we have the epithelial cells, these balls, and they sit on top of a mat of basal lamina, and underneath there is a ECM matrix of collagen, essentially the floor on top of which the cells sit. And this ECM is extremely important as it carries different source of critical developmental signals. So depending what kind of ECM there is, the cells will the cell fate of the cells will be determined by that. ECM is important for delivery of paracrine signals, as we saw. The secreted signals, sonic hedgehogs, winds, BMPs, activins, uh, RTKs, everything will be delivered via the ECM. So they do not just flow around in a liquid or something. These molecules actually travel along these ECM laminas, and it is critical for cell attachment. So they attach to the floor or ECM. Migration, they migrate on the ECM. Differentiation, so depending on what kind of ECM they face, cells may be change their fate or cause particular differentiations to occur. And finally, morphogenesis is also helped along by the ECM. So this is an example of how ECM cells bind the ECM. So one of the important proteins present in the ECM, and ECM is made out of mostly carbohydrates, uh, proteins which are modified with glucose or carbohydrate molecules, which are proteoglycans, and different proteins. So one of the important proteins are known as fibronectins. So fibronectins actually are bound to the ECM by covalent bonds, as we see here. There is a covalent bond right here. It binds to the ECM, and there are particular cell surface molecules known as integrins, which bind the fibronectin, which actually anchors the cell to the fibronectin, and in turn to the just give me a second. Like we lost power for a moment. It's coming back. Yeah. So integrins are cell surface molecules which are present on the cell, which bind to the fibronectin, which essentially anchors the cells to the ECM, because the fibronectin is bound to the ECM. So how does integrin binding work? They bind to the fibronectin via the RGD binding site, and these RGD binding sites are calcium binding sites. That is, the binding only works when there is calcium around. Calcium is required for the binding to take place. And on the intracellular side, the integrins via various different proteins such as tallins or vinculins bind to the actin microfilaments. So essentially they are anchored to the entire cytoskeleton of the cell. So actin essentially forms the entire cytoskeleton of the cell, essentially the framework on which the cell is standing. So a mesh of different proteins which makes the cell sturdy, or essentially it is essentially the bones of the cell, which causes it to maintain its shape. And via the integrin binding, the entire cytoskeleton is actually bound to the ECM. So essentially the cell is anchored to the ECM via this fibronectin, integrin, and actin binding. This is how it works. And the other thing that we need to know about is that which is epithelium to mesenchymal transition. So epithelial cells are the ones that stay and form the different linings in our body. So the intestinal lining, the blood vessel lining, everywhere we have a flat layer of cells. Those are epithelial cells which are anchored to the basement membrane or the ECM, and they are bound via the desposome complex and the integrins, anchoring them to the ECM. But then there are other cells which are known as the mesenchymal cells. These cells are not bound to the ECM and they can float around everywhere. And these cell types are interchanged between each other. So a epithelial cell can become mesenchymal due to some changes occurring. So what are these changes? So all of a sudden, if a cell decides that I don't want to be epithelia, don't want to be part of this layer of cells and bound to the ECM, I want to escape into other parts of the body. So it does something known as EMT, or epithelial to mesenchymal transition, in which after receiving particular signals or because of some changes occurring in the cell, it starts secreting something known as metalloproteases, or proteases that can chew up the connections between the cells as well as the connection to the ECM. So these proteases go and chew everything up, essentially making sure that the cell is now loose, and then the cell undergoes cytoskeletal changes to give up its shape and escape into the ECM layers, and essentially go from the epithelial state to the mesenchymal state. This occurs normally during development, especially seen in particular cells known as the neural crest cells, which are essentially cells from the top or the dorsal part of the neural tube, which undergo EMT or epithelial to mesenchymal transition to form the neural crest, and these cells then populate different parts of the body, forming various pigment cells, forming like traveling to different parts of the body, forming different structures. But EMT is widely known in case of cancers. In case of cancerous cells, these cells actually do something known as metastasis, where from the primary site of cancer development, cells start to metastasize and become mesenchymal and flow and go to different parts of the body and cause secondary cancers everywhere. So this is what happens in case of cancers as well, and this is known as metastasis. Okay, so coming back to the question now, which are the correct statements regarding role of extracellular matrix in development? A block in carbohydrate synthesis may adversely affect morphogenesis. This is true because ECM is made out of carbohydrates and protein modified by carbohydrates. With a block in carbohydrate synthesis, you do not have good ECM. So ECM, which actually causes morphogenesis via various methods, is inhibited because of this. Tissue scaffold treated with calcium chelators will prevent cell adhesion. That is also true, as we saw that the cells are anchored to the ECM and the fibronectin via calcium bound integrins. No calcium, no binding, so it is important for binding to the tissues, like tissues being bound together, and calcium chelators will just remove the calcium, and so you have no cell adhesion. And cancer metastasis requires ECM degrading enzyme. So the same thing, so, as epithelial to mesenchymal transition, it requires ECM degrading enzymes, and which is important in case of cancer metastasis. Okay, the next question is, an ERK dependent gene failed to express after induction with a specific ligand. Which of the following may be the possible causes? So we have already looked at ERK dependent gene expression in the RTK signaling pathway. Now you have to choose what might be the possible cause, even if even if the signaling ligand is present, why does it not activate? The options are overexpression of GAP, STAT5 failed to enter the nucleus, there was a failure in Hedgehog palmitylation, son of 7-less has a loss of function mutation, there is a gain of function mutation in GLI repressor, and LRP56 receptor has loss of function mutation in the ligand binding domain. Again, more than one option correct kind of a question, so go ahead and think what you think of what could be the correct answer. Okay, so let us look at it. We have looked at the RTK signaling pathway in the starting, but just refreshing your memory. So how does the RTK signaling pathway work? We have a ligand binding to the RTK receptor, which activates GNRP, activates RAS, RAF, MEK, ERK, transcription factor, and transcription. So in the ERK mediated pathway, so this is the path we are talking about. Just so we have discussed this, that GNRP is associated with the adapted protein son of 7-less in Drosophila, could be other adapted proteins, which in turn activate RAS by causing it to be bound with GTP instead of GDP, and then activate downstream signaling. And RAS GTP is regulated using the GAP proteins, which causes the GTP to be hydrolyzed to GDP, and then you do not have no longer have signaling. So what might be the possible causes of a ERK dependent gene to fail to express? Overexpression of GAP, as we have discussed earlier as well, could be a reason, because more GAP means that the RAS GTP gets hydrolyzed to the inactive RAS GDP, and thus you never have active signaling. Or the son of 7-less has a loss of function mutation. So what is son of 7-less? SOS is the guanine exchange factor, the GNRP, that adds the GTP to RAS. So essentially this protein which actually adds the GTP to RAS, activating it. But the GNRP is activated by the adapter protein son of 7-less. So if this is not there, there is no activation of GNRP and the entire cascade fails. So even if the ligand is bound, there will be loss of the there is no activation of the genes being transcribed by the pathway. So these two are the correct options. And the next question, select all the options that contain correctly matched pairs. So here we have on one end all the signalings we have been discussing: fibroblast growth factors, receptor tyrosine kinase signaling, which is the same thing. Fibroblast growth factors are a type of receptor tyrosine kinase signaling. JAK-STAT pathway, which is also a variation of receptor tyrosine kinase signaling but has specific roles. Hedgehog signaling, Wnt signaling, and here we have different roles that all of these signaling might play: thanatophoric dysplasia, feather development, limb formation, porcupine, monomeric G protein dependent, anterior posterior polarity, and cell cycle regulation. And we have been asked to actually do a match the following, and we have multiple options here. So I'll give you a second to look through it and figure out which options are correct, and it is more than one correct type of a thing. So it might be that you have to choose more than one option. So I will give you a second for it. Okay. So for quick reference, there is this table that you will obviously get when you get the PPT as well, in which we have particular pathways being related to particular roles, as well the core mechanism and key roles in development. So SGF pathway we have discussed causes thanatophoric dysplasia if we have a loss of function in it. RTK receptor pathway can also be related to thanatophoric dysplasia because it is just a subclass. FGF pathway is just a subclass of RTKs. JAK-STAT causes direct signaling to nucleus. Hedgehog helps in feather formation and as well as anterior posterior polarity. And Wnt beta catenin signaling causes feather formation as well as anterior posterior polarity. Hedgehog also helps in limb development as well. So going back, when we start to actually match fibroblast, so looking at the first option, A1, fibroblast growth factors, thanatophoric dysplasia, which is correct. B and 5, receptor tyrosine kinase signaling, monomeric G protein dependent. So RAS, RAF, these are monomeric G proteins as we have discussed. So yes, RTKs are related to this. Hedgehog and feather formation, as I just said, hedgehog is related to feather formation as well. And A3, fibroblast growth factor and limb development. We have seen that how it causes thanatophoric dysplasia by suppressing chondrocyte proliferation, so it is related to limb development. C and 1, JAK-STAT and thanatophoric dysplasia, again the same thing. And E and 6, which is Wnt and anterior posterior polarity. One of the key roles of Wnt is regulating anterior posterior polarity as well as cell proliferation. Okay, so this is all I had for you today. This is the reference that we have been following for so long: Introduction to Developmental Bio by Scott Gilbert, the 9th or the 10th edition, whatever you can get your hands on. The course has been taught on this, so I would recommend going through it once. and this is the YouTube link on which all the videos are being recorded and uploaded. So whenever you feel that you want to go ahead and have a look again, then you can just go to the YouTube channel. And thank you for watching the video, and I will see you next week. Thank you.